Welcome back to G Fun Facts Online, where today we are going to look past the headlines of genetic cures and dive straight into the biochemical machinery that makes them possible. We are talking about the safety of rewriting the human genome. Specifically, we are going to explore a breakthrough method called CHANGE-seq-BE, a tool that has emerged as the new gold standard for ensuring that when we edit DNA, we are only changing what we intend to change. If you follow science news, you know that the last decade has been defined by CRISPR-Cas9. We often call these molecular scissors because they cut DNA in half to disrupt a gene. But medicine is moving fast, and we are now entering the era of base editors. Think of these not as scissors, but as molecular pencils. Instead of cutting the DNA strand, they chemically convert one letter of the genetic code into another. This is huge, because over half of all known pathogenic human genetic variants, the errors that cause inherited diseases, are just single-letter typos. A base editor can, in theory, fix those typos without the chaotic damage of a double-strand break. But here is the catch, and it is a big one. Safety in theory does not equal safety in practice. The critical challenge remaining for the clinical translation of these base editors is the rigorous detection of off-target effects. These are unintended edits that occur elsewhere in the genome. If you are trying to fix a gene in the liver but you accidentally edit a tumor suppressor gene, you might cure a disease but cause cancer. We need to find those errors before they happen in a patient. This brings us to the topic of today's deep dive, CHANGE-seq-BE. Developed by researchers at St. Jude Children's Research Hospital, this method represents a massive paradigm shift in how we profile safety. It stands for circularization for high-throughput analysis of nucleus genome-wide effects by sequencing base editors. That is a mouthful, so we will stick with CHANGE-seq-BE. To understand why this method is so revolutionary, we first have to understand the safety paradox of base editing. Traditional CRISPR breaks both strands of DNA. When that happens, the cell screams for help. It triggers a massive DNA damage response. This makes off-target cuts relatively easy to find because they leave a scar, usually an insertion or deletion of code. But base editors are stealthy. They do not cut the DNA. They use a deaminase enzyme to chemically modify a base. For example, an adenine base editor, or ABE, converts an adenine to inosine, which the cell eventually reads as guanine. It is a silent edit. There is no scar. There is no break. Standard safety profiling methods that look for breaks are completely blind to these changes. Furthermore, base editors have a unique problem called guide-independent deamination. Sometimes, the enzyme just lands on the DNA randomly and changes a letter, even without the guide RNA telling it to. This is the invisible off-target problem. So how do you find a single changed letter in a genome that is 3 billion letters long? If you just sequenced the whole genome, you would be looking for a needle in a haystack, and you would need to sequence it hundreds of times to be sure the error was real and not just a sequencing glitch. This is where CHANGE-seq-BE comes in. It is an in vitro, or test tube, screening method. But unlike previous methods, it uses a brilliant biochemical trick to physically enrich the off-target sites. It effectively burns down the haystack, so you are only left holding the needles. Let's walk through the biochemistry of this step by step because the elegance of this protocol is what makes it the best in the world right now. The process starts with purified genomic DNA. In older methods, scientists would use sound waves or sonication to shatter the DNA into pieces, but that damages the ends of the DNA. CHANGE-seq-BE uses a custom enzyme called a TN5 transposome. Think of TN5 as a copy-paste tool. It cuts the genomic DNA and simultaneously pastes specific adapter sequences onto the ends. Once the DNA is cut and tagged, the researchers do something crucial. They circularize it. They use ligase glue to stick the ends together, turning the linear DNA fragments into covalently closed circles. This circularization is the key to the whole operation. Why? Because the background noise in sequencing usually comes from the ends of linear DNA. By turning the genome into circles, the researchers can then add a cocktail of exonucleases. These are enzymes that act like Pac-Man, eating any linear DNA from the ends inward. But because the circles have no ends, they are immune, they survive. This step ensures that we start with a pristine library of circular genomes with zero background noise. Now comes the test. The researchers introduce the base editor, the actual drug product being tested, into the test tube with these DNA circles. The editor goes to work, binding to its target, and inevitably, binding to its off-targets. It performs its chemical magic, converting adenines to inosines. But remember, the DNA is still a circle. It hasn't been cut yet. This brings us to the most ingenious step of the protocol, the endo-V reveal. The researchers add an enzyme called endonuclease V, or endo-V. This is a bacterial enzyme that specifically recognizes inosine, It ignores normal DNA, but when it sees inosine, it cuts the DNA backbone exactly one base downstream. This means that only the DNA circles that were edited by the base editor get cut and become linear again. All the unedited DNA, the vast majority of the genome, stays circular. Finally, the researchers do one last round of purification. They attach sequencing adapters to the newly cut ends. Because the unedited DNA is still circular, the adapters cannot bind to it efficiently. When they put this sample into a DNA sequencer, the machine reads primarily the DNA that was edited. This results in a signal-to-noise ratio that is orders of magnitude higher than simply sequencing the whole genome. You are getting pure signal. Now, you might be asking, how does this compare to other methods? Why not use digenome-seq or just test it in cells? Let's look at the comparisons. Genome-seq is another popular method, but it relies on sequencing the entire genome and using a computer to look for a pileup of reads at cut sites. It requires hundreds of millions of reads to see a faint signal. CHANGE-seq-BE physically removes the unedited DNA before sequencing. This means that 10 million reads on CHANGE-seq-BE can yield more data than 500 million reads on digenome-seq. It is faster, cheaper, and vastly more sensitive. Then there is the comparison to cell-based methods. You might think testing in live cells is better. The problem is efficiency. When you try to edit cells in a dish, maybe only 10 or 20% of them actually take up the editor. If you use those cells for safety testing, you are missing 90% of the potential data. CHANGE-seq-BE is biochemical. It saturates the DNA with the editor. It forces even the rarest off-targets to reveal themselves. It provides a worst-case scenario safety screen, which is exactly what regulatory bodies like the FDA want to see. Speaking of the FDA, let's talk about real-world applications. The release of CHANGE-seq-BE has already had ripples in the biopharma industry. A landmark application involved a potential therapy for X-linked hyper-IgM syndrome, or XIGM. This is a devastating immunodeficiency. Researchers had developed a specific adenine base editor called ABE8E to target the CD40LG gene. But before they could touch a patient, they had to prove it was safe. They ran CHANGE-seq-BE on the patient's own genomic DNA. The assay identified potential off-target sites. The researchers then took those specific coordinates and checked them in treated cells. They found no editing at those sites in the biological context. This was the proof needed to show that the risks were theoretical and not realized in the body. This data supported an emergency IND application, allowing the patient to be treated. In this context, CHANGE-seq-BE acted as the gatekeeper. It cast a wide net to find all possible errors so they could be ruled out one by one. But it is not just for safety checks, it is also for design. When scientists are designing a new therapy, they often start with 20 different guide RNAs. CHANGE-seq-BE allows them to screen all 20 in parallel. They do not just pick the most active guide, they pick the one with the cleanest profile, the one with the fewest off-target peaks. It is also used by protein engineers to build better editors. If a lab develops a new high-fidelity base editor, they can run it against the old version using CHANGE-seq-BE. If the new enzyme shows fewer peaks, it is objectively more specific. Of course, no technology is perfect, and we have to address the limitations. The biggest nuance right now is the mechanism itself. The protocol is highly optimized for adenine base editors because it relies on endo-V, which cleaves inosine. Cytosine base editors, which convert cytosine to uracil, require different chemistry. While the principle of circularization and enrichment applies, the current literature and protocols are heavily focused on adenine editors. There is also the discrepancy between the test tube and the living body. CHANGE-seq-BE is performed on naked DNA. In a living cell, DNA is wrapped around histones and protected by chromatin. This means CHANGE-seq-BE will almost always find more off-targets than actually exist in cells. But in the world of safety, this is a feature, not a bug. It is far better to have a false positive, a site flagged by the assay but safe in cells, than a false negative, where you miss a dangerous edit that ends up harming a patient. Looking to the future, the next frontier is integrating this biochemical data with biological context. Researchers are now looking at overlaying CHANGE-seq-BE results with chromatin accessibility data, like ATAC-seq. This would allow a computer to predict which of the biochemical off-targets are likely to be biologically active in a specific tissue type, like the liver versus the neurons. So for the researchers and students listening who want to implement this, the workflow is clear. You extract high molecular weight DNA, you tag it with TN5, you fill in the gaps, and digest the linear trash with exonucleases. You let your base editor do its work. You reveal the edits with endo-V, and then you sequence. It is a rigorous, step-by-step protocol that turns the invisible chemistry of gene editing into a digital signal we can analyze. In conclusion, CHANGE-seq-BE represents a triumph of biochemical engineering. As we move from the era of crude molecular scissors to precise molecular pencils, our safety tools must evolve with us. By converting the invisible chemical signature of base editing into a detectable, enriched signal, CHANGE-seq-BE provides the lens through which we can view the safety of the next generation of genetic medicines. It is the guardian of the genome, ensuring that in our quest to rewrite the code of life, we do not introduce unintended errors. Unbiased, high-sensitivity profiling is no longer optional in this field. It is the standard. And right now, CHANGE-seq-BE is the gold standard by which that safety is measured. Thanks for listening to this deep dive on Fun Facts Online.